Good morning, Ma'am Alma Patria. Good morning, classmates. And I am Leslie Pearl S. Bungkasan, and this is my video presentation about the microscope with its parts and functions. So before I proceed to my video presentation, I'll just read to you my objectives of this video. So this video presentation aims to let us know the different parts of the microscope and also to let us have the knowledge on how does each part of the microscope works. So what is a microscope? It is an optical instrument used for viewing small objects such as mineral samples, plant or animal cells typically magnified several hundred times. One way that microscope allow us to see smaller objects is through the process of magnification. So before we proceed to the parts of the microscope, let us recall who made the microscope. So a father-son team named Hans and Zacharias Janssen invented the first so-called compound microscope in the late 16th century. The first compound microscope dates to 1590, but it was Dutch Anthony van Leeuwenhoek in the mid-17th century who first used them to make discoveries. That's why Anthony van Leeuwenhoek named as the father of the microscope. When the microscope was first invented, it was a novelty item. So that is an example of a compound microscope. So the first part of the microscope I will discuss is the eyepiece or the ocular. This is what you look through at the top of the microscope. Typically, standard eyepieces have a magnifying power of 10x, and the optional eyepieces typically from 5x to 30x. So the next part of the microscope is the eyepiece tube, where it supports the eyepieces in place above and connects it to the objective lens. Next part is the arm. The arm is a support for the upper portion of the scope. It also serves as the convenient carrying handle. The next one is the base. If the upper portion of the microscope has a support, therefore, the bottom of the microscope has a support too, and that is the base. The next one is the illuminator. Illuminator is the light source of a microscope, typically located in the base of the microscope. So the next one is the objective lenses. The objective lens gathers light from the specimen, magnifies the image, and projects the magnified image into the body tube. 4 separate lenses that magnify the image: 4x, 10x, 40x, and 100x, depending on the objective in use. The lens is positioned just above the object being viewed. The next one is the coarse adjustment and the fine adjustment. So coarse adjustment is a rapid control which allows us for a quick focusing by moving the objective lens or stage up and down. It is used for initial focusing, while fine adjustment, a slow but precise control used to fine focus the image when viewing at the higher magnification. To make it short, coarse adjustment is used only for lower magnifications, while fine adjustment is used for higher magnifications. The second to the last part of the microscope is the rack stop. It is a safety feature that prevents the viewer from allowing the objective lens to accidentally hit the stage and damage the specimen or slide. And last but not the least, the condenser and iris diaphragm. Condenser is used to collect light and focus light from the illuminator onto the specimen. It is located under the stage, often in conjunction with an iris diaphragm, while iris diaphragm controls the amount of light reaching the specimen. It is located above the condenser and below the stage. So that's the parts and functions of the microscope. So the conclusion of this video presentation: microscopes have been long an important instrument in the medical field. The world-changing discovery of germ theory, which saved countless lives, was made possible because of microscope. As a medical technology student, the microscope is an important element of our education, and by understanding its parts and function, we will be able to utilize it properly. That's all. Thank you for listening. Bye-bye!